Hello class, we would like to start a lecture on major histocompatibility. When we talk about major histocompatibility, we are referring to the fact that one individual differs from another individual. Even Siamese twins, identical twins, they differ. So what causes these significant differences among these people? That's the question we should ask. Some people in humans, major histocompatibility complex. In humans, major histocompatibility complex is called human leukocyte antigen. Human leukocyte antigen. You realize that some people get infected by parasitic infections. Just a small dose and those people are done. Others too, no matter what malaria or HIV will do to them, they become carriers. What is the significant differences between all these people's genome or their genetic makeup? Is the major histocompatibility. Polymorphisms in this major histocompatibility can lead to disease dispositions. It can also lead to organ transplant rejection or graft rejections, because if the molecules of major histocompatibility are lower in an organ, meaning that that organ, when you transplant that organ from a donor to a recipient who has low major histocompatibility complex or MHC, then there is reduction in graft rejection. Let's go to the classroom as we start the lecture. But know that this is our last but one lecture. The last lecture will be on hypersensitivity reaction, but we need this foundation to be able to establish the link between major histocompatibility and delayed hypersensitivity type 4. Thank you. So, when we talk about major histocompatibility complex. Okay, so the lecture seeks to give an overview of the role of the major histocompatibility complex in immune response. We will also be describing the structure and function of class 1 and class 2 MHC molecules. We will discuss the nature of polymorphism in class 1 and class 2 MHC molecules. So let's begin. Let's begin with a video.
In the last video lectures we covered antigens. We studied that antigens induce an immune response inside the body. Most of these antigens are large protein molecules. Some are polysaccharides and a few are glycoproteins or nucleoproteins. We also came to know about self-antigens or autoantigens. Among these self-antigens, the antigens which are found on the membranes of almost all the cells of human body and other vertebrate animals are known as major histocompatibility antigens. They are glycoproteins in chemical nature. Major histocompatibility antigens are also known as histocompatibility antigens. These antigens were discovered when scientists were doing transplantation experiments on mice. Scientists found that sometimes the transplant tissue from the donor mouse was accepted by the recipient mouse, but at other times it was rejected by the recipient mouse. Now the question was, why the tissues from one individual of a species were destroyed when introduced into a member of the same species. The answer was given by an American mouse geneticist, George Snell. He gave the reason that this rejection was because the tissues of the donor and the recipient were incompatible. In other words, it was discovered that if the animals were not closely related, the recipients rejected the grafts. So, if the donor mouse and the recipient mouse are of the same genetic background or same inbred strain, the transplanted tissue is accepted by the recipient. However, if the donor and recipient mouse are of genetically different backgrounds or different inbred mouse strains, the transplanted tissue is rejected by the recipient. Further research revealed that a group of closely related genes was the cause of the rejection. They named this cluster of gene the major histocompatibility complex, abbreviated as MHC. The major histocompatibility complex got its name from the fact that the genes in this region encode proteins which determine whether a tissue transplanted between two individuals will be accepted or rejected. So, it was discovered that these genes encode proteins which act as self-antigens on the surface of the cells. These proteins are known as major histocompatibility antigens. These antigens gives the answer why rejection of the transplanted tissue happened. When a tissue was transplanted from donor mouse to recipient mouse, the major histocompatibility antigens of donor mouse were perceived as foreign or non-self by the recipient mouse. As a result, the immune system of recipient mouse mount an immune response and result in the destruction of the transplanted tissue. Let's understand some important points about major histocompatibility complex. If we look at the term histocompatibility, histo means tissue and compatibility means getting along or agreeable. The MHC got its name from the fact that the genes in this region encode proteins that determine whether a tissue transplanted between two individuals will be accepted or rejected. The term complex represents that these genes are localized to a large genetic region containing multiple loci. Since the molecules encoded by these genes were found to have main effects on histocompatibility, to distinguish them from other molecules encoded by genome having minor effects on histocompatibility, they were called major histocompatibility molecules. And the genes encoding these molecules were called major histocompatibility complex. Major histocompatibility complex is defined as a tightly linked cluster of genes whose products play important role in intercellular recognition and in discrimination between self and non-self. Major histocompatibility complex are present on chromosome 6 in humans, and they are known as human leukocyte antigens, abbreviated as HLA complex. In case of mice, they are present on chromosome 17 and are known as H2 complex in mice. MHC molecules are found on all nucleated cells in the body, and they play an important role in the development of both humoral and cellular immunity. We know that B cells react with antigen on their own. But T cells recognize antigens only in peptide form, and that too when combined with an MHC molecule. So, the main function of MHC molecules is to bring antigen to the cell surface for recognition by T cells. In humans, the genes coding for MHC molecules are found on short arm of chromosome 6. The MHC molecules are divided into 3 classes, namely Class 1, Class 2, and Class 3. Class 1 molecules are coded at 3 different locations or loci, termed A, B, and C. These glycoproteins are expressed on all nucleated cells. Class 2 genes are situated in the D region, and there are several different loci, known as DR, DQ, and DP. These glycoproteins appear only on cells that can process non-self materials and present antigen to other cells. The area between the Class 1 and Class 2 regions on chromosome 6 is of Class 3 genes. These genes code for complement proteins and cytokines which take part in immune response, but these molecules are not expressed on cell surface.
I guess you all watched the video. So, I guess you all watched the video. Yes, so the video explained what major histocompatibility is. And in humans, it is found on the shorter arm of chromosome 6. The shorter arm of, I've forgotten the chromosome. So let's continue. But in rats, it's in chromosome 6. Let's continue. Okay, so the major histocompatibility complex is a large genetic complex with multiple loci. Genes in the MHC were first identified as being important genes in rejection of transplanted tissues, meaning that at first, when you have somebody has kidney failure or liver failure, how do you help that person so that the person can survive for some number of years before dying? When something like this happens, then if the person is coming from a rich home, then those people will go to an organ bank. Some of these banks are located in India, in South Africa, and in Europe, depending on how much the organs sell. And these are organs that have been donated by people who were about to die, so that the organs will be kept in a bank, so that when somebody comes to buy some, they will use it to take care of those people's families. and those organs are so expensive. Imagine a liver can cost over billions and billions of cedis, because there's nothing you can equate life to. A rich man would like to buy with such a big money so that he can survive for some number of years. So that's why we have what? Organ transplants. But with organ transplant, we have realized that if the genetic makeup of the donor and the receiver are not equal or are not similar, then there will be what? Host versus what? Versus graft rejection. Host versus what? Graft rejection. So you see that the person, the donor and the receiver, their organ, the organ of the donor will be incompatible to the immune system of the receiver. So there will be rejection. The receiver is going to produce antibodies against the donor's organ, and this will elicit immune response, which is very, very devastating and very, very worrying. Okay, so genes within the MHC were highly polymorphic, meaning that people in Mali will have different major histocompatibility genetic makeup than people in Niger or in Ghana. Studies with inbred strains of mice showed that genes within the MHC were also involved in controlling both humoral and cell-mediated responses. The major histocompatibility complex, or whenever you hear MHC, is a large genetic. There are 3 kinds of molecules encoded by MHC. So the major histocompatibilities are 3. We have the class 1, class 2, and class 3. The MHC products play roles in intercellular recognition and discrimination between self and non-self. So the reason why your antibodies or immunoglobulins will not get up one day and say, we are going to fight and destroy this person's eyes. Your own antibodies will rise and say, let us destroy your eyes, let us destroy your liver, let us destroy your what? And it depends on the major histocompatibility. That is making it impossible for you yourself, your own antibodies or cells, to fight against you. The MHC participates in the development of both humoral and cell-mediated immune response. While antibodies may react with antigens alone, most T cells recognize antigen only when it is combined with an MHC. So, T cells, before they can recognize an antigen, it has to be bounded to what? An MHC. So we have MHC1, MHC class 1, MHC class 2, MHC class 3. MHC class 1, MHC class 2, MHC class 3. The class 1 is found in all nucleated cells. All nucleated, that means that cells that are having nucleus. All nucleated cells. Whereas the class 2 is found in, so like I said, the MHC class 1 is found in all nucleated cells. For instance, the cells of the heart. The heart has what? Cells like this. We call them the cardiomyocytes or cardiac cells. Cardiac cells. That's the cells of the heart. Cardiac cells have what? MHC molecules on their surface. Let me draw it with the red. MHC molecules on their surfaces. MHC. Class 1 molecules on their surface. So MHC class 1. Liver cells have, let me draw a liver cell. Let me bring the green. So the liver cells MHC are here. This is MHC for liver cells. So that's class 1. So liver cells are called hepatocytes. So you can see that even MHC class 1 is also found on white blood cells surface, lymphocytes. It is also found on cancer surfaces of what? Cancer cells. Okay, MHC class 2 is only found in what? Found on antigen presenting cells. On antigen presenting cells. So an antigen presenting cells, examples are macrophage, dendritic cell located in the skin, and lastly we have the basophils. We also have the neutrophil. We have the, so these are antigen presenting cells. So let's see on the surface of the antigen presenting cell. Let us see what we want to see. So antigen presenting cell, let's say if this is macrophage, that is the very big cell. Macrophage, and this is the nucleus for macrophage. So MHC molecules are found on the surfaces of what? MHC class 2. The first one we did is class 1. So this one, class 2, are found on the surfaces of what? All antigen presenting cells, like the dendritic cell and the macrophage. So this is MHC class 2. MHC class 2. MHC class 2. Then this one, the function of the MHC class 3 is that it produces complement proteins, like the complement cascade. This MHC class 3 provides the proteins, and some are also saying it produces the cytokines and other things. So let's leave it at the protein. We may come and refer later. Okay, so this is what we have for complement. This is what we have for the MHC. So let's go back to our slides. Okay, so class, I've already explained everything, so the rest I'm just going to read because that's what I've explained. Class 1 MHC genes encode glycoproteins, and these are expressed on the surface of nearly all nucleated cells, but vary in concentration on different cell types. So like I said, we are on the class 1. The class 1 MHC is on what? All nucleated cells. Example, the hepatocytes, the cells of the eye, the cells of the kidney, that is the cells of the kidney, the cells of the blood, the connective tissue, the cells of the liver, the cells of the liver, the cells of the spleen. They all display MHC class 1 on their cell surface. They all display MHC class 1 on their cell surface. Okay, so let's go. So major function of class 1 gene product is presenting peptide antigens to cytotoxic T lymphocytes. So let's see class 1, what work is it doing? So class 1, the work it will do is that it will present, I guess you understand how I can clean it. So class 1 will present antigens. So the class 1, let's see, this one has the class 1 MHC. Class 1 MHC, it has a receptor. So we have this is the antigen, because it will swallow the antigen by phagocytosis. Then later it will display it on the surface, on its receptor surface. Then this after displaying it, so this is MHC1, MHC class 1. Okay, then this is the antigen. Like I said, these ones are expressed on the surface of all nucleated cells. So that is as expressed. Then cytotoxic T lymphocyte will come and also bring its receptor and come and join to come and digest the antigen. This cytotoxic T lymphocyte, it will bring its antigen and come and feed on, it will come and feed on the antigen. So this is what cytotoxic T cytotoxic. Then this is the first one is what? Any cell. So we can say liver cells, liver cell, liver cell. My liver is not well spelled. Liver cell. So you can see this liver cell is a nucleated cell. It has a nucleus. So it represents the antigen on this red thing is called MHC class 1. It's a receptor. So it will now hold the antigen and present it to this macrophage. It will present it to this cytotoxic T lymphocyte. And you know what cytotoxic T lymphocyte? It will release. It will feed on the antigen straightforward. Class 2 MHC genes encode glycoproteins expressed primarily on antigen presenting cells. I've explained that one. Macrophages and the antigen presenting cells, we have the macrophages, we have the dendritic cells, and we have B cells. We don't have neutrophil. It's the B cells. I made a mistake. Where they present processed antigenic peptides to helper T cells. Okay, so let's see. So for MHC class 2, we have MHC class 2. It uses what? Antigen presenting cells. The class 1 uses nucleated cells, but the class 2 uses what? Antigen presenting cells like the macrophage. So the macrophage will swallow the organism up. Then after swallowing it up, it will display its antigen on its surface. It will display its antigen on its surface like this. So where is the antigen? Here, antigen here, antigen here, antigen here, antigen here. And this antigen that's the the MHC. This is MHC class 2. This receptor is called MHC class 2. Class 2. So now, afterwards or after this, then this one works with what? The class 2. The class 2 MHC works with what? T helper cells. So let's see, T helper cells will come down here. That is its nucleus. Then we have the MHC molecules here. T helper cells has what? Receptors. So it will bind onto the antigen. It will bind onto the antigen. After binding onto the antigen, there will be a series of signal transactions that will lead to the production of what? That will lead to the cytotoxic T helper. This is T helper cells. This one will lead to T helper cells. T helper cells producing cytokines. So T helper cells will now produce cytokines that will destroy. They also produce lymphokines. This one will destroy the parasite or the antigen. So the lymphocyte, the cytokines should be produced in large quantities, and it will go and destroy the antigen and kill it. This one too, it will go and destroy the antigen and kill it. Okay, so class 3 MHC genes encode various secreted proteins. So like I said, class 3 MHC gene produces what? The complement system, and also is involved in inflammation. So this is the structure of major histocompatibility complex. Another structure. In humans, the HLA gene is called, the MHC gene is called HLA. That is human chorionic gonadotropin. No, sorry. In humans, the MHC gene is called MHC gene in human is called human leukocyte antigen. Human leukocyte antigen, or HLA for short. This, instead of MHC, we rather have HLA in human beings. So these are the morphologies. There are also HLA-E, HLA-A, HLA-F, HLA-E, HLA-F, HLA-G. Because of polymorphism, because there's supposed to be only HLA-A, B, and C. But now, because of mutations and genetic manipulation and other things, we have HLA-E, HLA-F, HLA-G. Cellular distribution of MHC molecules. In general, the classical class 1 MHC molecules are expressed on most nucleated cells, but the level of expression differs among different cell types. The highest levels of class 1 molecules are expressed by lymphocytes. So lymphocytes express higher level of class 1 molecules. In contrast, fibroblasts, muscle cells, liver hepatocytes, neural cells express very low levels of class 1 MHC. I have already explained, especially the classical class 1 MHC molecules are expressed on most nucleated cells. I've already explained this. So the low level on liver cells, the low level of the MHCs on liver cells may contribute to considerable success of liver transplant by reducing the likelihood of graft recognition by the cytotoxic T cells, by the cytotoxic T cells of the recipients. I've already explained this. I said that when the MHCs are different genetically, then there is going to be host versus what? Graft rejection. There's a high likelihood of the graft being rejected by the host because the donor and the recipient are not the same. They don't have the same genetic makeup, and even their genetic makeup are not related. They are not so close. So the tissue will be rejected. But in case the MHC levels are MHC level is an established fact that the MHC molecules on liver cells are very few and scanty. So when you present an organ to them, maybe a kidney, somebody donates his kidney to somebody who has low level of MHC on his liver, then the kidney will live or stay with the liver forever. There will not be any reaction. The body will not produce any antibodies against the kidney, because genetically the donor and the recipient are similar. Unlike class 1 MHC molecules, class 2 molecules are expressed only by antigen presenting cells. I've explained this. Primary macrophages, dendritic cells and B cells, and thymic epithelial cells. I explained how class 2 molecules display the antigen on the surface of macrophage or antigen presenting cells, so that in the end such an allergy can be taken out of the body by the macrophage. So in the end, such an organism can be taken out from the body and destroyed by what? The T helper cells producing cytokines. But and also note that MHC class 2 molecules mediate the production of cytokines by T helper cells to destroy the antigens. So MHC class 2 molecules bind onto what? T helper cells, and T helper cells produces the cytokines which destroys the antigen that has been displayed on the surface of the antigen presenting cell. So let's see. Class 1 MHC is for nucleated cells. Please learn this very well. Know the type of cells that class 1 MHC is found on. Then class 2 MHC is found on antigen presenting cells. That means that they are on the macrophage, they are on the B lymphocytes, and they are also on the dendritic cell. We would like to end our lecture on major histocompatibility here. Let's now take a break as we come back to continue with hypersensitivity reactions. Thank you.